Well, welcome everyone. It's absolutely a pleasure to see you here for the College of Medicine's Dean's Distinguished Lecture. I'm absolutely honored to have Dr. Charles Rice with us here today to share his presentation, A Cure for Hepatitis C, Now What? A seemingly simple question, which I'm sure has twists and turns and complexity in terms of the answer. Again, thank you so much for being here, and thank you for joining us, all of you in the room, and those joining us in a simulcast at our other campuses in Rockford and Peoria. We launched this Dean's Distinguished Lecture Series in the fall of 2022, as the pandemic was waning, to highlight research topics that integrate the efforts of multidisciplinary teams. As we all know, biomedical sciences has become a team sport and requires different subject matter and technical expertise, including cell biology, molecular biology, systems biology, and so many more. I'm sorry if I forgot your particular area of interest. Um, Given this reality, it's essential to expose our junior faculty and really our senior faculty as well, as students, residents, and fellows to lectures and seminars on research topics that integrate efforts from multidisciplinary teams that have worked together to solve major scientific problems. The critical objective of this lecture series is to engage our College of Medicine broadly with leading edge science that involves both the depth and breadth of scientific knowledge and technical expertise. These lectures lead to further insights into building successful scientific teams and new ideas about diversifying and expanding our research programs at all levels. We are so pleased to be hosting Dr. Rice, who, of course, has made seminal contributions in our understanding of hepatitis C. And these basic understandings have led to translational impacts and clinical impacts that have affected the lives of millions of people. I'd also like to thank our Vice Chancellor for Health Affairs, who's here, Dr. Bob Barish, who's been very supportive of this lecture series, as well as Chancellor Miranda, who isn't here right now, but I think will be here a little bit later for the reception. So with that, I'm now pleased to introduce Dr. Susan Ross, the Sweeney Professor and Head of the Department of Microbiology and Immunology, who has been important in getting Dr. Rice here and who has the privilege of introducing Dr. Rice. Dr. Ross studies the genetics of host virus interactions, particularly retroviruses and New World arenaviruses. She is an outstanding scientist, is elected fellow of the American Academy of Microbiology in 2002 and a AAAS fellow in 2009. Please join me in welcoming Dr. Ross.
So I'm really happy to be able to introduce Charlie Rice today. Charlie is the Maurice and Corinne Greenberg Chair in Virology at Rockefeller University. I won't list the other titles because they're up there on the screen. Charlie got his PhD, his bachelor's degree from University of California in Davis, and his PhD from Caltech in 1981, where he studied the alpha virus, Synbis virus. Not that many people study that, but it's a mosquito-borne virus that's actually very important in human health, and also began to study flaviviruses, which particularly at that time, the yellow fever virus. And he studied a strain of yellow fever that was later used to develop the yellow fever live attenuated vaccine. He then moved to Washington University to begin his faculty career, which is where he started his work on hepatitis C virus, another flavivirus. This is for the virologists in the audience. And really made amazing discoveries about the biology of the virus, how it replicates, how its RNA is used for replication. And it really led to the ability to produce the first infectious molecular clone of HCV, which was critical not only to understanding the biology of the virus, but for setting up culture models and in vivo models, non-human primate models that could be used to develop antivirals. And as many of you now know, that about, I think it was like 10 years ago, that the first antivirals against HCV became widely available in humans. I actually have a little bit of a personal story about that. My first lab manager had to quit working because he developed liver cirrhosis from HCV infection and unfortunately passed away about 5 years before the development of the drug. So I always feel a little bit sad when I hear about this amazing discovery. So Charlie has authored over 500 articles in the field of virology. He served as a reviewer and editor for many, many journals. He is the past president of the American Society for Virology, a fellow of the AAAS, a member of the U.S. National Academy of Sciences, a recipient of many prizes, including the Lasker DeBakey Prize, and of course, the Nobel Prize in Medicine in 2020, along with Harvey Alter and Michael Houghton for the discovery and development of HCV. And I think in addition to all these accomplishments, an amazing thing about Charlie is the number of fellows and students he's trained. When I looked at the list of fellows and students and saw who was on that list, and there are over 65 postdocs and 20 grad students, I recognized many of their names because they populate microbiology and infectious disease departments, as well as industry positions all over the world. So with that, I'll let Charlie.
Well, thank you to the Dean and to Susan for sort of that sort of wonderful windup. Got quite a crowd here today. Thanks to you all for coming. And it's a real honor to be here. I very much enjoyed my day sort of meeting with colleagues and with students. And we had a fantastic lunch where I got to sort of learn some more things about hepatitis B virus that I didn't know. So it's really been a wonderful day. So this is going to be kind of a sort of a historical talk, sort of that is centered around hepatitis C. And I'll start off with just a little bit of sort of ancient history. And certainly hepatitis has been around for a long time, and there are sort of records of this, back into sort of the ancient Greeks and Romans. And outbreaks of jaundice, particularly when lots of people got together in sort of wartime activities. In the 1930s, when the yellow fever vaccine was developed, it was actually sort of stabilized with human serum. And some of those recipients of the vaccine in that period of time actually developed hepatitis, but it wasn't a hepatitis that looked clinically like sort of yellow fever. So this really sort of got at the idea that, sort of maybe there was a link between sort of autologous serum, whether it be human serum into humans or animal serum into animals, that sort of was associated with hepatitis. So in the 40s, I think it became widely recognized that there were two types, sort of this infectious versus sort of serum transmitted. And in 1965, this is a picture of Bloomberg here, sort of discovered hepatitis B virus in blood. Baruch won the Nobel Prize for that sort of discovery some years later. And then in 1973, this shows you Bob Purcell, Al Kapikian, and Steve Feinstein around a rather old version of an electron microscope that used immunoelectron microscopy to sort of basically identify the hepatitis A virus particle. So during this time, it became possible to sort of determine, sort of whether an infection was due to hepatitis A and hepatitis B. And in the mid-70s, it was noticed that in the transfusion setting, there were still cases of hepatitis that were not due to hepatitis A or hepatitis B. And this was really sort of the observation of Harvey Alter at NIH and Fred Prince at the New York Blood Center. And they termed this post-transfusion non-A, non-B hepatitis, kind of a somewhat vague name. And this wasn't a rare occurrence. It occurred in about 10% of transfused patients. And it resulted in persistent hepatitis, which could develop into advanced fibrosis and cirrhosis, and later was shown to be associated with the development of liver cancer. It was transmitted to chimpanzees, which really formed the sort of cornerstone of characterization of this agent. And basically, the chimpanzee model was used to show that this was, in fact, an envelope virus. It could be basically inactivated by treating it with sort of organic solvents. So this observation, and really not knowing if this was sort of one agent or many agents, sort of led from the mid 1970s to really sort of figure out, sort of whether this was one agent or sort of multiple agents that were responsible for this condition. And this took almost 15 years in order to sort of get to the finish line. And in 1989, Michael Houghton and his colleagues, along with contributions by Harvey Alter, sort of published this paper in Science, where they were able to use expression cloning of a cDNA library using convalescent serum to, identify a little piece of this non-A, non-B sort of viral sort of genome. And then they could sort of build out this sequence. And that gave rise to, basically the non-A, non-B agent being called hepatitis C virus. So at this time, I was at Washington University. I was like sort of 3 years into starting my lab. And one of the things that this abstract from Michael's paper said was that this is an RNA molecule, the genome of this virus. It's positive stranded, and it was like, potentially the sort of togaviridae, which were sort of envelope viruses like Symbis, or the flaviviridae, like the sort of prototype flavivirus, yellow fever virus. So that was of interest to me because in 1989, we were spending a lot of time working on yellow fever. And it is, I mentioned, the sort of prototype member of this virus family. And the live attenuated vaccine strain, which was developed by Max Tyler in the mid 1930s, is one of the most safe and effective vaccines that's ever been developed. It's been in about 500 million people, one shot, lifelong protection. So we were very interested in trying to figure this out. And we published the sequence of this virus in 1985 when I was still sort of at Caltech. And then the real driving force was to really try and create a genetic system. And I'll say a little bit more about this later, but in order to study these positive strand RNA viruses, it's not like working with DNA. You've got an RNA genome. So how do you do site-directed mutagenesis on an RNA genome? Well, the way that positive strand RNA virologists do that is to actually force it through a cDNA intermediate so that you can actually sort of have the genome in a plasmid, for example, and you can make whatever changes you want, transcribe an RNA and transfect it into cells and see what the phenotype of that mutant might be. And in this case, we were very interested in the case of yellow fever to be able to actually potentially use this vaccine strain to not only understand why it was attenuated and so effective, but also potentially use it as a carrier for epitopes from other pathogens. So this was a situation so that we were sort of working in 1989. This paper published in the New Biologist, a up-and-coming journal which went extinct shortly after this paper was published in it. Anyway, that's where we were in 1989. And so I was in my office at Washington University, and I got a call in the lab from Steve Feinstein, who I didn't know. Now, as I mentioned before, Steve had been one of the co-discoverers of hepatitis A virus. And he was now at the FDA and also had a laboratory there. And he was very interested in developing a vaccine for this non-A, non-B hepatitis C, sort of viral agent. So he had seen our paper on yellow fever and he thought, well, no problem. We can just sort of take the 8CV antigenic, sort of glycoproteins and stuff them into sort of yellow fever 17D, and voila, we'll have sort of a vaccine that might protect against both yellow fever and HCV. And in talking about this, we thought, well, it's a great idea, but at that point, we really didn't know much about, sort of hepatitis C, how the proteins were expressed. So that really, I think in that era, sort of 1989 and the early 90s, it became clear that this non-A, non-B agent was a huge problem, and that it was distributed throughout the world with some areas that were, pretty hot, sort of like in Egypt, for example, but also in the United States, estimates of more than 100 million people that were chronically infected with this hepatotropic virus. And it was pretty clear that this chronic infection, which occurred in a majority of those that were infected, regardless of immune status, sort of could progress to these different stages of liver disease from, basically just inflammation in the liver, hepatitis, to a much slower trajectory to cirrhosis and end-stage liver disease, including hepatocellular carcinoma. So this was, a significant public health problem to, basically have 100 million people basically cooking with this sort of chronic viral disease. And at the time, there really wasn't much in terms of what could be done in terms of HCV treatment. People started to treat patients with type 1 interferon, which had been used for hepatitis B virus before. And, that worked, but it had significant side effects, but it only worked in about 5% of those that were treated. And they had to basically take the systemic interferon for 6 or 12 months or even longer to get to that sort of poor response rate. So I think, sort of at the outset of this new virus, sort of being on our radar screens, cleaning up the blood supply was done relatively quickly, sort of with, very sensitive diagnostic to detect HCV in the blood. High-risk groups, we really sort of needed to sort of educate them and hopefully develop a vaccine. We have not managed to do that yet. And in terms of, sort of successful treatment, in order to, treat people, you have to identify those that are infected. And it turns out that for hepatitis C, in many people, it's a subclinical infection. So they can be infected with this virus and it can be cooking away for decades and they don't even know they're infected unless they would have, for instance, a blood test that would reveal an elevated liver enzyme. So improving treatment was, definitely sort of on the radar. And the ultimate goal is and still is to eradicate this virus. So the problem with hepatitis C was that it didn't replicate in cell culture. And there was no small animal model, and we could only use the chimpanzee model, and that model actually hasn't been possible to use now for almost 10 years. So a lot of the early work was done using the chimpanzee model, but as you can imagine, the sort of number of animals that you could use for these kinds of experiments was sort of very limited. And, that was a very important model, but not one that, most labs could embrace. So, sort of here I was in 1989 trying to decide whether or not to work on this virus. And as Susan's mentioned, we had worked on SINVIS. I told you about our work on yellow fever and sort of thinking about a scorecard, genome sequenced, yes. Infectious clone, we had that for SINVIS and yellow fever, not for HCV. Growth in cell culture, great for SINVIS, a little less well for yellow fever, no way for hepatitis C. Animal model, there were all animal models, but I've told you, at least for HCV, it was the chimpanzee. Robust lab effort that was ongoing was, we were doing a lot on SINVIS and yellow fever, nothing on HCV. Tools and reagents, again, sort of a dearth of these for HCV. And important infectious disease, well, SINVIS maybe not quite so much, yellow fever to some extent, and HCV, as I've just told you, sort of a very sort of important widespread disease around the world. So, the question was who in the lab would be crazy enough to study HCV? There were a couple of us. This is sort of, you may not recognize me in this picture, but the other guy is Arash Grakui, who is the sort of technician that I first hired when I went to Washington University. He's now a professor at Emory. And we decided to, begin studying this virus kind of as a side project. And so the early days were taking sort of what we knew, sort of about the HCV genome and trying to understand a little bit more about, sort of how the virus expressed the proteins it needed in order to replicate. And this schematic sort of shown here, basically shows you an internal ribosome entry site that this virus doesn't have a traditional 5' cap, and then a long open reading frame. And then this long open reading frame encoded, and Arash worked on this, 10 different proteins that were basically produced by a combination of cellular proteases, host proteases, and 2 viral proteases. And even early on, before we could culture this virus, there were clearly enzymatic activities that were predicted from the sequence. People succeeded in developing biochemical assays for those enzymes. And this is the NS34A protease helicase and the NS5B, the last protein in this polyprotein, which is the RNA-dependent RNA polymerase. So these are kind of, favorite sort of targets for drug development. But at this point, there was no way to actually tell what we were understanding from these surrogate systems to study HCV if they were really important in the context of the virus life cycle, because we didn't have a cell culture system where we could probe their importance. So that really sort of brought up this effort to, sort of make a functional molecular clone for hepatitis C. And, normally you would just have a virus, permissive cells, mix them together, and you get more virus. Simple. All the herpes virologists and other virologists in the audience are pretty spoiled by this. But for positive strand RNA viruses, you can extract the genome RNA, and if you have a permissive cell, you can bypass the entry process, transfect it into cells, and if everything is there in order to replicate, it will, basically kick off replication and make progeny virus. You can then make a cDNA copy of that viral genome. You can engineer it, basically so that you can transcribe essentially unlimited quantities of the genome RNA of the virus, transfect that into cells and get virus. So we embarked on, sort of trying to do this for hepatitis C. And the question was, what are we going to use as a starting material? It turned out that Harvey Alter had a transfused patient at the NIH where he actually collected some acute phase plasma. And this patient was called patient H, and the date of the acute phase collection was 1977, so 877 patient H. And the reason this seemed like a good substrate to start with was that it was basically a very highly infectious acute phase serum sample, and we knew that because it had actually been titrated by limiting dilution in the chimpanzee model. So we knew that the specific infectivity of this material was like, 10 to 100 genome copies would initiate an infection in the chimpanzee model. So pretty good material. So we put together with Steve Feinstein and others in my lab sort of a prototype 8-strain cDNA sequence. We transcribed that with RNA to sort of make our genome RNA mimic. We tried transfecting that into a bunch of cell cultures. I told you there's no sort of validated cell culture for, hepatitis C at this point, but we thought that maybe that could be an entry block. So if we could transfect in replication competent RNA in those cells, that, it would be able to replicate. That didn't work. And so we were sort of left to the only validated model was the chimpanzee model. So how do you test in sort of a naked in vitro transcribed RNA to see if it could initiate infection in a chimpanzee model? You think this is just nuts, right? You've got this 10 kb RNA, one nick by a ribonuclease, it's dead. And you're talking about actually delivering this to an in vivo sort of animal liver model, sort of like the chimpanzee. But it turned out that Sue Emerson and Bob Purcell's lab had actually succeeded in doing this in marmosets for hepatitis A virus some years earlier. And so we went ahead and gave it a shot, and it didn't work. So that was pretty disappointing. You're going to hear a lot of these sort of disappointing things along the way here. So we began to wonder as to whether or not the actual hepatitis C sequence that had been reported was actually a complete sequence. And that was because the Chiron sequence from Michael Houghton's group actually reported that this had a poly-A tail like cellular messenger RNAs. And the work from some groups in Japan with a different sort of subtype of genotype 1, genotype 1b, were finding these sort of poly-U, polypyrimidine tracks at the 3' end. But we knew from our work on yellow fever that this virus, which is again a relative, but a distant relative to hepatitis C, actually had a rather stable stem loop RNA structure at the 3' end, which was very difficult to clone. And so I was really fortunate to have Sasha Kolokolov in the lab. Sasha came from the Soviet Union and sort of immigrated to the United States. He was really already, when he arrived in the lab, a superb sort of molecular biologist. And he took, sort of very limited amounts of this 877 plasma that we'd have. We're talking literally microliters of this stuff, and worked out, what people today would say is a conventional 3' race protocol to sort of clone the 3' ends of RNA molecules, and managed to succeed in identifying a sequence that had been missed at the 3' end of the HCV genome. So this was done simultaneously by Kunatada Shimatono in Japan for a genotype 1b isolate. And basically what you can see here is from the stop codon of this long polyprotein that I pointed out, there's sort of a variable region among HCV subtypes, an internal poly-U polymyxin tract of sort of varying length, and then this sequence of about 98 bases that turned out to be essentially the most highly conserved RNA element in the HCV genome. And we know that these RNA viruses are very highly adapted and evolved. You can't just lop off 100 nucleotides of conserved sequence and expect them to work. And so we were convinced that this was really sort of the sort of missing piece. So we basically, sort of tacked it on to our sort of prototype Hutchinson strain clone, transcribed RNA, cell culture failed again. And then in the chimpanzee model, again, intrahepatic injection of this transcribed RNA into the chimpanzee model, that also failed again. So again, we thought we had an aha moment, and yeah, it didn't work out that way. So, what could be going wrong? Certainly delivering the RNA by just injecting it into the liver and hoping that it's going to be able to get across a cellular membrane, get translated, and initiate replication was asking a lot. Could there be part of the genome still missing? We didn't think so because we worked pretty hard on both the 3' end and the 5' end. Maybe this wasn't a conventional positive strand RNA virus, and maybe you actually needed a protein or an RNA modification that would only occur in the context of replication, but not in our sort of in vitro RNA transcription reactions. But the other thing that we thought about that we could actually address was whether or not, sort of maybe our clone had, lethal variations in it, and so it was non-functional. And there were a couple of potential sources of that. One was the sort of incredible heterogeneity that this virus generates during chronic replication in an individual. I mean, it's estimated that there are about 10 to the 12th variants generated per day in a single chronically infected individual. So there's a lot of variation. Some of those are going to be tolerated, and some of them are going to be probably deleterious. In those days, sort of the enzymes involved in cDNA synthesis and cloning were not as good as they are today. So certainly errors could be introduced during that. So we decided to actually take the sequences that we had, which were derived from multiple clones, and basically create a potential clone that had the consensus nucleotide sequence at every position in the HCV genome. Now, we didn't know if this consensus sequence actually was present, because I mean, this was kind of cobbled together, but we figured at least that was a good way of filtering out sort of potential sort of lethal mutations. And so Sasha, again, sort of put this together, which was really an amazing tour de force with the tools that we had at the time. And lo and behold, this actually worked. And this is two chimps that were transfected with this RNA. And I have to say that after all of the experiments that we'd done, and Steve Feinstone at the FDA and NIH was doing the chimpanzee work, we didn't have funding to do that. Every time that we tried this before, there was essentially no RNA signal in the circulation, days or even a week after sort of administering lots of RNA to the liver. And so it was really amazing when we sort of had the first report that there was HCV RNA circulating sort of in these animals and increasing over time. And this just shows you in blue, basically the increase in viremia over time. In red, this is a liver enzyme that's a marker of liver damage that usually sort of kicks in 2 to 3 months after hepatitis C infection. And essentially, so these chimpanzee infections mimicked sort of what we see with sort of experimental infections in chimpanzees and also acute infections in humans. So this was really sort of a wonderful sort of step forward, and we were, feeling pretty good about it for a couple of reasons. One is that it really sort of formalized that hepatitis C alone was sufficient to actually initiate this infection and cause disease. So this is kind of the sort of molecular equivalent of Koch's postulates. So I don't think that we really had too much doubt at that time that, sort of what had been sort of discovered was the causative of this, but this was sort of formal proof. And, we could begin to do clonal infection in the chimpanzee model, look at host, sort of response and evolution. We could test the importance of drug targets, so we could actually sort of mutate some of these enzymatic activities and show that they were required for replication. But now we were able to actually sort of produce unlimited quantities of this, basically in vivo validated RNA. So you'd think you could take this and, put it into every different kind of cell culture system that you could think of and find, something that would work. And unfortunately, that didn't work. So that was, again, sort of a step forward, but, sort of not where we wanted to be for this virus. So the next breakthrough was from Volker Lohmann and Ralph Bartenschlager in Germany, and they were working with a genotype 1b isolate. And I think we were all, sort of really wondering, sort of where was this field going to go if we actually couldn't sort of manipulate these viral genomes and to study their function. And what Volker and Ralph did was to create what we call a subgenomic replicon. So it had the internal ribosome translation initiation site at the beginning, and then they put a cassette for neomycin resistance, basically another internal ribosome entry site to basically drive production of what were felt to be the proteins required for RNA amplification. So if this is transfected into cells, if it can replicate without killing the cells and express this drug-resistant marker, you should be able to get some neo or G418 resistant colonies growing up. And that's what they found. In a human hepatoma cell line, HU87s, a few colonies sort of grew up sort of under drug selection, and they had persistently replicating subgenomic sort of HCV RNAs in those cells. The problem was that only about 1 in a million transfected cells actually, sort of had this phenotype. So it really wasn't a workable sort of genetic system to sort of work with this virus. But it turned out, and we in other labs, sort of found that, and again, this was a very important advance in terms of having sort of the first cell-based system. But in order to make it better, it turned out that if you actually isolated the RNA from these rare colonies and sequenced it, it wasn't what we started with. And in fact, there were changes that basically allowed this to replicate in cells. So if these were re-engineered, sort of back into the original replicon, some of them could increase the replication efficiency of this by more than 50,000-fold. So really a huge, and then we could also cure the cells, sort of with interferon, for example, and then isolate subclones of these HU87 cells that were more permissive. So at the end of this, we really had a genetic system where we could begin to sort of, sort of study the replication of this virus, do genetics. This was extended to other HCV genotypes. There are 6 major genotypes. We could engineer reporters, and this really sort of became the driver of drug discovery. So you would think that, okay, we've got this thing, the self-replicating RNA in cells, perfectly happy. It's expressing a drug marker or reporter genes. What about just sort of translating that and trying to get a complete replication cycle? So we and others, sort of introduced these adaptive mutations sort of into our chimpanzee, sort of validated infectious clones, transfected this into HU87 cells. These RNAs replicated and produced the proteins that we thought were important, and no virus was released. So again, it sort of seemed like here we had sort of everything replicating in these cells, but, sort of no infectious virus that could be transmitted to other cells. So the next saga here was from Takashi Wakita in Japan, and Takashi was interested in, trying to get a cell culture replication system going. And he thought that maybe by sort of studying HCV isolates from these very rare patients that had acute fulminant hepatitis due to hepatitis C, might have, sort of variants that had sort of more robust replicative capacity, and they would sort of be better at, creating a good cell culture system. So he had one of these called Japanese fulminant hepatitis 1, JFH1, which he basically engineered a replicon of the type that I just showed you. And this particular replicon had the ability to establish robust replication with high efficiency in cells without any adaptive mutations. And so at the time we were thinking that, sort of maybe these adaptive mutations and sort of these other isolates and genotypes were great for encouraging sort of non-cytopathic persistent replication, but they were not so good in terms of later steps in the virus life cycle to actually produce virus particles. So I was fortunate to have sort of two sort of postdocs in the lab, Brett Lindenbach shown here on the left and Matt Evans. And so we began to look at this along with Takashi Wakita in the Bartenschlager lab and Frank Cesari at the Scripps. And basically we found that this Japanese fulminant hepatitis isolate, just either the sort of wild type sequence or some chimeric sort of constructs that we made that were even more efficient, could actually make virus. So this was in 2005, and we could show that this virus could not only sort of be used to infect sort of naive uninfected cells and produce more virus, but it could also infect chimpanzees, and from those we could actually recover tissue culture infectious virus and also humanized mice. These are mice that have been engrafted with human hepatocytes, and basically it's a model where we can actually create about 90% of the human of the hepatocytes in the liver being human. So this was great. And I see this is a movie, a live imaging sort of movie. And I just sort of, this is basically on the left, we're looking at the accumulation of HCV antigens over time from sort of this initial infection event. And on the right, this is actually an indicator where the hepatitis C viral protease cleaves a reporter protein off of the mitochondria that then relocalizes to the nucleus. So we could actually sort of use this to actually finally, sort of study this virus in various ways. And so it really, sort of from that time on, we really sort of had a lot of tools to study this virus. And I can't sort of review, sort of everything that we've learned about the life cycle and virus-host interactions and immunology, and also to look for sort of other viruses in nature. And I'll say a little bit more about that later. And now I think hepatitis C is, sort of one of the best studied RNA viruses. Certainly not up to the standards set by SARS-CoV-2. That's sort of changed dramatically. So I'll say a little bit about treatment, because I mean, that's where, sort of all of this was heading. I mean, for HCV, as opposed to HIV, for example, we have several things sort of going for us, although at the time, I don't think that we really, other than the data from interferon, where we knew that a cure could be, at least a virus elimination could be obtained, we didn't really know if there was going to be something that would, really be able to cure this virus in a broad spectrum of the infected patients. But the genome, it's a cytoplasmic RNA virus. It doesn't integrate in the host cell nucleus. Even the interferon era had shown us that if you cure the chronic viral infection, it lowers your risk of sort of progressive liver disease. So that's good. And that if you really sort of achieved what we call the sustained viral response, which is virus negative 6 months after cessation of treatment, the virus is pretty much gone. So it's, even if people become immunosuppressed later on, it's not like the virus, comes back. So that was good. And these are the sort of 3 targets that sort of emerged as the winners. You can imagine that sort of with the advent of the replicon system, which was really focused on the nuts and bolts of RNA replication, those targets really sort of achieved a sort of front and center attention. I mentioned the NS3,4A protease and the RNA-dependent RNA polymerase before, and then there was a multifunctional protein called NS5A, which compounds were identified that inhibited this by basically an unbiased small molecule screen. And it turns out that it was additional med chem. Those compounds, many of those compounds are incredibly potent sort of antivirals. as potent as anything that's been seen so far. And this protein is involved both in RNA replication and virus assembly. So, you know, it's really amazing now that we have these treatments that we have today. So it's basically 2 to 3 months in most cases of treatment, 1 pill a day, essentially few or very few side effects, manageable drug-drug interactions in some cases. And these cocktails that consist usually of 2 of these direct-acting antivirals, or in some cases 3, actually target, you know, sort of all of the major HCV genotypes. And there are 6 of them, as I mentioned before. These are quite variable. It's really, I think, to me, sort of amazing that the drug discovery industry has been able to, you know, fashion these compounds, you know, to sort of deal with this kind of sort of genetic heterogeneity. So it's kind of a rare occurrence for, you know, sort of a scientist to sort of be involved in kind of the trenches of this and sort of see this development, you know, sort of going from this mystery virus in the 1970s with a 5% cure with interferon, the identification of the virus after heroic effort some years later. The first direct-acting antivirals were added to interferon and ribavirin therapy in 2011, but that was really kind of a mess. This was a very toxic combo that was better at sort of curing the virus, but, you know, patients sometimes had to be hospitalized, and I can see some people shaking their head in the audience. But it was in 2014-15 when we could actually get rid of interferon and largely get rid of ribavirin and achieve almost a complete cure for this virus infection. So there are challenges that remain. Who's infected, as I've mentioned. This is often a silent infection, so there are still a lot of people that don't know their infection. It's hard to sort of implement sort of these advances. I think as a basic scientist, you think, oh, you know, all these great new drugs, flip a switch, identify everybody, they're cured. This is going to be dragging on for a while, and that's true sort of even in the United States. Affordable treatment has been an issue. Challenging patient groups, not so much. With the sort of interferon therapy, you know, people with HIV, HCV co-infection, which is quite common, sort of were more difficult to treat. People with more advanced liver disease were more difficult to treat. With these direct-acting antiviral cocktails, not so much. Viral resistance isn't too much of a problem, and we still don't have a vaccine yet. So, I mean, this, you know, sort of, we're talking about, you know, sort of 8 years ago or so, you know, sort of what am I supposed to do as a molecular virologist? Continue or abandon basic work on HCV, tackle other viruses. We certainly have done some of this, hepatitis B, yellow fever, Zika, and SARS. Build on the HCV work, but diversify, that's it, all of the above. I think that's the right answer, and then maybe consider an alternative career. That's probably what I should do, although maybe it's a little bit late. So, I mean, we still don't have a vaccine, and I think the success with COVID-19 has provided some enthusiasm for trying to go after that. And we still understand very little about sort of disease progression of HCV. I mean, how does this virus infection lead to, you know, sort of fibrosis, cirrhosis, and cancer? You know, is this an effect of just chronic inflammation, or are there other viral determinants that are important? So in the case of HCV, as I've sort of mentioned before, if you get rid of the virus, it lowers your risk of developing liver cancer, hepatocellular carcinoma. So that's great, and it probably means that, you know, we should aggressively treat as early as possible. The bad news is that even in cured HCV or suppressed HBV, those are still risk factors for developing liver cancer. So I think there's definitely impetus to sort of understanding, you know, sort of more about this and how to sort of detect early cancer and come up with better treatment. So even if you get rid of the virus, particularly if you have advanced liver disease, you're not out of the woods in terms of developing liver cancer. So one of the things that we've been trying to fill this gap is to, you know, the chimpanzee model. We can't use it anymore. It was super important in sort of all hepatitis virus research. but we really need a better model to address some of these fundamental questions. And you can try and adapt the virus. So far, that hasn't worked to sort of produce, say, a virus that can replicate efficiently in mouse cells. You can identify the factors that sort of make this sort of uniquely human specific and basically transplant them into mice, which we've done. And we can actually make a mouse that's permissive for HCV infection, but it's basically a sort of a low level infection that, basically fades out and is without sort of any pathology. And then the other was to, basically find a related virus in nature that we could use as a model to understand some of these processes. So it turned out, let's see, what did I want? It turned out that I have a friend at Columbia, Ian Lipkin, who's kind of a virus hunter. And he's been, sequencing, deep sequencing samples from all kinds of different animals. And every once in a while, I'd get a call on the phone from Ian, and he'd say, well, Charlie, we discovered a new hepatitis C related virus. Do you want to work on this? So the first one was a equine virus, so a horse virus. So horses are really not welcomed on the Rockefeller campus. But we started working with this virus in collaboration with the vets at Cornell and Ithaca. But it was clearly, it was great in the sense that there was no problem with getting large sample sizes. We've been worrying about microliters in a mouse. You can get liters from a horse. But anyway, that wasn't really sort of what we were looking for. The next phone call came with a deer mouse, Paramiscus. And so he had discovered, again, sort of some new hepativiruses in deer mice. So we got these deer mice, which, sort of look very cute as far as mice go. And yes, they could be chronically infected with this virus, but sort of no pathology. And it turned out that, unbeknownst to us, but we found out later, none of the immune reagents that we use for working on laboratory mice, sort of work with this deer mouse. They are phylogenetically so far away that perhaps this shouldn't have been a huge surprise. I mean, we tried taking this virus and putting it into every immunodeficient laboratory mouse that we could find. No luck. So then there were, sort of relatives that were found in bats and also relatives that were found in gibbons. None of those were really sort of models. And then the last phone call that I got from Ian on this, which really has been, very interesting, had to do with his teaming up with some pest control groups in New York City. And what they did was to head out there and they trapped a bunch of Norway rats that were sort of scampering around sort of Central Park and other places in New York, and basically sliced and diced and sequenced these guys. And they found two lineages of Norway rat hepativirus in these animals. So, we got the virus, and, basically Eva Bellerbeck, sort of a postdoc immunologist in the lab, asked, well, will this rat virus infect laboratory mice? And Eva found that yes, sort of we could infect sort of laboratory mice with what we call NRHV, Norway rat hepativirus. And Eva, basically showed that we could actually develop a chronic infection model, because traditional C56 black 6 mice, it's an acute resolving infection in 2 to 3 weeks. If you deplete CD8 cells before infection, it's slightly delayed in terms of clearance. But if you transiently deplete CD4s, you can establish a chronic infection in these animals. And that is maintained even after the CD4s come back. And it turns out that this model actually does cause pathogenesis in these animals. So I'll show you some of the data, but I think the most exciting finding was that the 100% of the male mice, after sometime between 12 and 18 months post-chronic infection, develop liver cancer. And the female mice, it's about half that frequency. And we see, without going through the details, some of the same sort of liver pathology that we see in chronic HCV and HCV-associated HCC. So we're really excited about this. This is new. We actually haven't published this last part that I sort of mentioned about this. And I think it's going to be giving us a chance to sort of look at mechanisms of oncogenesis. We're interested in these sort of sexual dimorphisms that we see between the sort of male mice and looking at other comorbidities, sort of like diet and fatty liver, alcohol, and age. Sort of is the fibrosis that we see actually important for cancer development in these animals? We don't know that. And there actually may be ways of sort of blocking or reversing fibrosis progression. And the impact of virus cure on fibrosis. I mean, I sort of mentioned this at sort of the beginning of this section, that in the case of chronic HCV, if you treat, in some people, it seems that, basically the die has been cast for developing liver cancer. Sort of when does that occur? Sort of how long after chronic infection? So we're sort of figuring out ways to sort of cure chronic infection. This gives us a model to actually sort of look at different kinds of therapies for pathogenesis. And the other thing that's kind of interesting is the role of host genetics. And maybe Susan will sort of appreciate this, being sort of a mouse person, but there is a resource which is called the Collaborative Cross. And these are basically mice that are inbred strains, but they're very, very different from each other. So if you have something that you're interested in, and let's say if you take, sort of 10 of these Collaborative Cross founders and you infect them with virus X or whatever, and you see sort of different phenotypes, then you can actually use mouse genetics to sort of figure out, sort of the locus that's involved in those phenotypes by making F1s and F2s and then genotyping them and phenotyping. So Tim Sheehan, who is a former postdoc in my lab, who's now a faculty member at UNC, has been doing this. And basically, he's identified a collaborative cross strain, CCO70, that basically establishes chronic infection without this sort of immune manipulation, and sort of use the difference between Black 6 and that strain to begin to sort of focus in on what the host determinant is that's responsible for that. So anyway, that's sort of what I wanted to sort of conclude with today in terms of, in terms of the sort of acknowledgements for this NRHB work. We've been at it for several years. We're still trying to convince the NCI to fund it. That better happen soon. And Mariana Batista, a postdoc from Brazil, has really been the one that has led this. And it is a sort of an experiment that we started in earnest about 6 months before the pandemic hit, to sort of collect 6, 12, and 18-month time points. And so it was just a nightmare at this supposed 6-month time point. And so, in terms of hepatitis C, it has been sort of a long road, and it's been traveled by many. I think when you get sort of recognized with these awards, I just want to emphasize that, I only had a little bit to do with this success story. And the people in the lab, colleagues, people all over the world, physicians, people participating in the clinical trials, those were all, sort of very important components to sort of making this a success story. This is a word cloud of sort of my sort of HCV collaborators sort of over the years. Steve Feinstone gets, working with him and having the ability to do these chimpanzee experiments was absolutely essential. We've had a great support staff through the years, both at WashU and at Rockefeller. And I really like this quote from Lewis Thomas, which is somewhat dated, but I think is true today as it ever was. It says, in the course of scientific progress, things tend to pop up. Most of the celebrated achievements had their origin in moments of surprise in the laboratory, unplanned for, unanticipated, unpredicted. No committee convened to survey the future prospects for biomedical research could possibly have guessed at the things that lay ahead. No branch of government could have laid out detailed plans for the scientific needs of the future beyond asserting that what the country needed was more fundamental knowledge about the human form and function, about the agents and influences responsible for disease, and in a certain sense, about nature itself. Scientific research works and is the only way to get at the underlying mechanisms of disease and the only way to learn what we can do about them. So I think, that's sort of a message for everybody in this room. I think it's very important. And I guess the last one would be this one. If you think something is important, just, sort of don't give up. Thank you.
A couple of minutes past the hour, but we'll entertain two questions. Two questions. Who wants to go first? If there aren't any, then we can adjourn and continue the discussion at the reception. Any questions? Oh, I'm sorry.
I can shout.
Yeah.
So just what do you think about the prospects of eradication of HCV in the future? And what would you think to get there?
Well, that's a great question. I mean, I think the most important thing is to have, a will to sort of achieve that goal and identify the means to do that and implement them. We've only done sort of a partial job in the United States, and I think in terms of, sort of the percentage that are still chronically infected or acutely chronically infected is going up, not down. So we need to do a better job of identifying infected people and treating them. I think the cost has come down, so that's good, but it's still not cheap. I think the other example of what can happen, and maybe this will become, sort of a model for us and other countries in the world, is in Egypt. In Egypt, about 10% of the population of Egypt was chronically infected with hepatitis C, genotype 4. And this happened because of a schistosomiasis sort of campaign in Egypt where people were lined up and basically administered this cocktail with a syringe that was not cleaned up in between participants. So Egypt really had this sort of brewing problem, of liver disease. And they had actually sort of managed to negotiate a deal with one of the pharma companies producing these DAAs, Gilead, to produce the drug in Egypt and basically administer it at a price that their public health system could afford. And in the last, sort of 5 years in Egypt, they've successfully eliminated more than 90% of chronic HCV infection. So I think it can be done, but it's going to take sort of a combination of factors to sort of make it happen. So I think it is possible, but it's still going to be, given the complexity of global infectious disease, it's going to be a while before it happens.
Well, with that, let's thank Dr. Charlie Rice once more, and we'll see you on the steps.